All right, so this is part 2 of our podcast on our kidney outlines. Hopefully last time you got a clear view of the parts of the kidney and kind of an intro to the parts of the nephron or the functional unit of the kidney. So remember, if we're looking at the nephrons, there's millions of them in each kidney. And kind of these dots represent these parts of the nephron up here, and then the lines represent these parts. And we have longer and shorter nephrons. Some of them dip way down into the medulla, and others are shorter. So this picture is pretty realistic on the different types of nephrons. About 20% of our nephrons will dip all the way down into the medulla, and those are important in maintaining water balance. So if we look at this nephron system, we notice that there is definitely blood very close to kidney parts. So, you know, one thing you might do on your worksheet is actually take like a yellow highlighter and yellow highlight all of these parts or tubes that are yellow in this picture, as I think that helps you to visualize that a little better. And then if you want to color red and blue the other tubes, I think that's also helpful. So continuing on and looking a little deeper into our nephrons, so this next page here where it says nephron structure off to the side. So the parts of a nephron, we're just going to start where it connects closely to the blood supply, is the part called the Bowman's capsule or the glomerular capsule. And so this is this cup-shaped structure that we see right here wrapping around that glomerulus. So we've got the glomerulus is that ball of capillaries that came into the kidney region, went in through the afferent arteriole, through the ball of capillaries, and out through the efferent arteriole, and it created that great pressurized system so that we can have filtration occurring here. So our Bowman's capsule is this cup-shaped structure with a highly porous inner wall. It collects fluid filtered from the blood. Then as we travel down, so this is kind of a blow up of our nephron again. So we just talked about our Bowman's capsule. And then, and you can kind of, you know, picture that like your hand. And so the glomerulus would be, kind of think of it like a baseball. The glomerulus would be the baseball and the Bowman's capsule would be the glove or the cup. And then the cup continues down, and I can't morph my wrist, but we would see next the proximal convoluted tubule. Okay, and so proximal, it's closest to the point of attachment. Right, remember that term from first chapter? Convoluted, it's all warped around in a variety of ways, and it's a tubule. So it's a highly twisted section of the nephron with cells in the wall having mini mitochondria and microvilli projecting into the lumen of the tubule. And remember, lumen is the center of the tube. So they look a little like this. And you see kind of this fuzzy lining on the inside. So if you're looking at kidney tubules, the cells of kidney tubules are not squamous, not columnar. They are cuboidal, simple cuboidal epithelial cells. So you see these beautiful circles when you look at kidney tubules. So you see these cuboidal cells. They're about as wide as they are tall. And then we've got the microvilli in the lining. So then it goes from the proximal convoluted tubule down to the loop of Henle. I've always thought the loop of Henle was fun. So as we saw in that other picture, some loops are long and some are short. And we're going to talk a little later about, you know, in different animals, what the difference is in those two. So the loop of Henle has this hairpin turn. Consists of a descending limb where it's traveling down away from the Bowman's capsule, and then an ascending limb that brings the filtrate back into the cortex. So descending limb goes to the medulla, ascending limb back out to the cortex. So the next section, go back to the picture here, would be then the distal convoluted tubule. So our distal convoluted tubule is another highly twisted section. It doesn't have as many microvilli, and it doesn't have as many mitochondria. And then it dumps then into the collecting duct. So back to our picture here. So it's going to dump into our collecting tubule or our collecting duct, and so a variety of nephrons would dump into that same duct. So it's a wider tube, carries the filtrate back through the cortex and the medulla to the renal pelvis. So if we look at, again, our kidney tissue, again, our distal convoluted tubule is going to be a little wider, more open looking than what our proximal convoluted tubule is. Our PC, or proximal convoluted tubule, has this microvilli brush border. So one of the main reasons, and we'll talk about this more later, that the proximal convoluted tubule has that microvilli lining or layer is because that's the majority of reabsorption that occurs there. So think about, how we had the microvilli in the small intestine, so we're increasing surface area, and so it's going to allow for greater reabsorption. So looking at the basic principles in urine formation, maintaining homeostasis is definitely going to be key by regulating the volume and composition of blood, and it gets rid of metabolic wastes. So the three most important metabolic wastes that the kidneys excrete or get rid of from the body are urea, which is produced during the breakdown of amino acids, creatinine, which is generated in skeletal muscles by the breakdown of creatine phosphates, uric acid, which is formed by the recycling of nitrogenous bases from RNA molecules. So that all those extra bases. So really there's three major actions that occur in the kidney to allow for urine formation. So the first one is filtration, and so filtration occurs between that glomerulus and Bowman's capsule. And we talked about the fact that the capillaries in the glomerulus have a lot of open pores in them. They're more open to allowing things to come out. So these fenestrae, or pores in the wall, will increase fluid transfer. We talked about that increased pressure in the glomerulus because of the big afferent arteriole sending blood in and the small efferent arteriole taking blood out. So most solutes will freely filter out, but the proteins are retained in the capillaries. So big things like all the blood cells should stay in the capillaries, any large proteins that are floating around in the capillaries, some of the polypeptides, but most of the other things will filter through that system. So the fluids or particles pass through the pores or the fenestrae, and then they go through the basement membrane, and then they pass these little structures called podocytes, and podocytes are just little feet-like structures that are part of the glomerular capillary. So the picture I've put on your sheet there, if we take just a little cross section of a glomerular capillary and blow it up, we see the little podocyte foot, and you see them kind of blown up here around the glomerular capillary. So the key reasons for those podocytes is really they're structural. They're going to provide support to that capillary. This capillary has all of these open pores, so it's a little bit delicate. So those podocytes are going to give the support that that capillary needs to allow it to be able to filtrate like it does. And I think we're going to pause there to and so watch podcast 3 also, please.